So our next speaker is Susan Ellenberg. Dr. Ellenberg is professor of biostatistics at the Department of Biostatistics and Epidemiology at the Perelman School of Medicine at the University of Pennsylvania, and her research is focused on practical problems and ethical issues in designing, conducting, and analyzing data from clinical trials, including surrogate endpoints, data monitoring committees, clinical trial designs, adverse effect monitoring, vaccine safety, and special issues in cancer and AIDS trials. Prior to her appointment at the University of Pennsylvania, she held positions of increasing responsibility in the U.S. federal government and the NIH and the FDA. So she'll bring a perspective from both sides here, and she's going to speak to us about what we've learned about designing, conducting, and interpreting randomized trials over the past years. Thank you.
Thank you. Thank you, and I'd like to thank the organizers and Dave in particular very much for inviting me here. This is a very interesting set of discussions, and I'm taking lots of notes. My disclosure, I do have a lot of other activities, but I don't see any of them having any potential for conflict of interest with this presentation. So, the modern era of clinical trials. There's been debates, controversies constantly over the last 65 years. I think many of them have been resolved, at least in general principles, that randomization is valuable, it's an ethical thing to do, that we can terminate trials early without inflating our error, that placebo controls are okay in certain situations. That's a relatively recent resolution, I think. I hope it's resolved. Others continue to inspire debate. We don't have a lot of consensus on how conservative we should be about stopping trials early. How do we prevent ourselves from being misled by multiple comparisons? What do we do? The Bayesian versus frequentist discussions. And I think we've seen that the wheel is regularly reinvented. Adaptive designs are all the rage now, but people were talking about it. Clinical trials have always been adaptive in some sense, and there have been adaptive designs forever. Same with pragmatic trials. I have the Schwartz and Leloup book on my shelf that I think was published in 1960 on explanatory versus pragmatic trials. So, but again, this is the latest thing. So the randomization, I think when it was put forward and first used in the late 1940s, it was adopted fairly rapidly, but there were certainly vocal dissenters, particularly in the cancer arena, who said things like, we know that our current treatments are lousy. And these new treatments are, have so much promise. I mean, look what they did to the mice, So how can it be ethical to give half of our patients treatments that we know are unsatisfactory, they're going to die? Everybody should be able to get the new treatment. And there was a lot of passion about this. And certainly in areas where there is no standard treatment, we regularly see this argument come up. When I was, shortly after I went to the FDA, my part of the FDA was working on a trial of a new treatment for ALS. There were no treatments for ALS. The primary outcome of this trial was delay and worsening of symptoms, and the trial was designed as a placebo-controlled trial, and the patient community was so unhappy that they actually were able to have a 60 Minutes production where the FDA was viewed as just terrible for allowing this to go on. The patient said, we have 2 years, we can't wait for a resolution. In this case, the trial actually did go forward, was stopped early because the drug arm was doing worse. That did not get onto 60 Minutes, although I urged Janet Woodcock to call Mike Wallace and demand a rehearing. The concerns about randomization and ethical issues spawned approaches to maybe make randomization improve the chances that people would get a better treatment. Zelen in 1969 proposed the play the winner design, which is a very simplified version of a response adaptive randomization. Bill Rosenberger is sort of the guru of this area. Lots of methods, earn methods, with the possibility that as a treatment becomes looking better, more people will be randomized to what appears to be the better arm. But although this sounds very good, it really hasn't caught hold, and I think it's probably due to a number of reasons. Others of you may have other ideas. To me, one is that the study is designed in a way that says we know that one treatment is looking better, but you're still randomizing some people. You're still getting some people who are getting something that the study design has already said is inferior. We may be creating convenient fictions when we do clinical trials that we have zero knowledge of which is better at the beginning, and then the knowledge is a step function. We have zero knowledge as the trial goes on, and at the end, we have the information, so then it goes up. It's a convenient fiction, but it makes me a little more comfortable. These designs also require larger sample sizes, so it's not entirely clear in some situations that you actually have fewer people, maybe have a lower percentage, but maybe not fewer people getting the inferior treatment. And of course, it requires the assumption of no time trend in prognosis, which can be problematic. The issue of patients as willing participants has an interesting history. The Declaration of Helsinki was put forward first in 1964, which specified that informed consent had to be given for non-therapeutic research, but did not require it for therapeutic research, as consistent with patient psychology, is what the original Declaration of Helsinki said. And then we had the very famous Henry Beecher article in the New England Journal of Medicine in 1966 that documented lots of experiments that were widely viewed as unethical, upset a lot of people, and led to the first requirements for informed consent, both from NIH and FDA. The National Commission was established. The Belmont Report was issued in 1979. Now, Bob Temple, who looked at my slides, tells me that I'm wrong, that GC was not done without participant consent. I know I read that somewhere, so maybe somebody will enlighten me about whether, in fact, there was informed consent in the GC trials. But certainly there were some large trials done without informed consent. But the concerns about the consent process and the extent to which the requirements for consent cause problems in getting important information has carried on, and I think there have been some changes. Zelen, again, in 1979, proposed the randomized consent design in which people would be randomized between a standard therapy and a new therapy, and proposed that people getting the standard therapy in these trials did not need to be consented because they're getting what they would otherwise have gotten. I'm not sure that's possible now within the current regulations, but that was a proposal, and not sure any trial was ever actually done. Well, actually, there was one. The ECMO trial was done with this design, where the children assigned to standard treatment and did not get ECMO were not told that they were in a study, but it hasn't been done very often. In 1991, HHS in the United States authorized IRBs to waive consent requirements in some circumstances. So again, sort of recognition that maybe there's some cases where we can get by without consent. And in 1996, after a lot of intense discussion, FDA and HHS collaborated on changing, on adding regulations that permitted the conduct of certain emergency research studies, like the CRASH studies, without patient or proxy consent. So today, we continue to have these debates, as we just heard in the last talk. We have concerns about the length and readability of consent forms. Are people really being informed? Many of us are quite concerned that they're not. The debates about need for consent in the kinds of studies we just talked about, cluster trials, particularly those to compare standard treatments or to assess quality of care. Most of us recognize that much about the consent process is driven more by legal considerations than really informing patients, but people haven't figured out how to roll this back very well. So placebo controls. Initially, before the era where people were widely given informed consent, there was concern about deception, that you're going to give people a drug and you're going to tell them that it might help them. Of course, in many instances, it could, but that you weren't telling them that they might be getting a placebo. But there's been a chronic tension about the ethics of withholding therapy, and in the 1990s, there was a huge escalation of this debate when Ken Rothman and Karen Michaels first published their paper arguing that the Declaration of Helsinki said that placebo controls were always unethical when there was existing treatment, no matter what you were treating. You have Viagra, so therefore it's not possible to test Cialis in a placebo-controlled trial. Not sure that got too much traction, but a few years later, there was this huge debate about the placebo-controlled trials of prevention of mother-to-child transmission in developing countries after a regimen had been shown to be highly effective in the United States. The trials done in developing countries were done with placebo controls because the regimen that was being administered there was vastly simplified from what was done in the United States study and was felt to be could not be delivered in environments where there was no standard prenatal care, and a concern if they compared to the best treatment available anywhere may very likely be worse, but you wouldn't know whether it was good at all. So there was lots and lots of debate about this and led to multiple revisions of the Declaration of Helsinki, which went from saying absolutely you can never use placebo controls to sort of kind of going back to now saying specifically, yeah, you can use placebo controls as long as they're needed scientifically and nobody's going to come to any major harm. So one lesson I learned was that when you talk to people about ethics, and if they have a strong feeling about something that they think is based on ethics, they really don't care about the science. And I went to several meetings on this where you could see people like this with their hands over their ears. You know, don't talk to me, don't tell me about it. I know it's unethical, and that's all we wrote. But this one has come out, I think, in a sensible way. However, the issue of how you do trials with active controls, which we often do, has been complicated. It's very natural to test new against available therapy. As the 60s and 70s and 80s progressed, there was growing recognition that not being able to show a statistically significant difference between A and B did not imply that A and B were equivalent, and that specifying a difference to be ruled out, which was one step up, that was not sufficient, that one also needed to establish the basis for that difference and look at the variability of prior results. We have now seemingly settled on the phrase non-inferiority trials for such studies. That's not really a very good word, but that's what we're calling. I think people are understanding this better, but there's still a lot of problems. I remember, I call this a light bulb moment because I was at this advisory committee. I was working at the Cancer Institute then, and I was at an advisory committee meeting that was looking at a new drug for breast cancer. And the results of several trials comparing the new drug to the standard drug were presented. In each of these cases, the new drug looked worse in each trial. The differences didn't meet statistical significance, and the difference in response rate was within the amount, I think, that had been pre-specified. I think it was 15 percentage points for how much worse the new drug could be. And Tom Fleming, who was then a young statistician at the Mayo Clinic, was the statistician on the advisory committee, and you could kind of see, you could really almost like see the light bulb going on, and he noted that the confidence intervals around the differences could not exclude the possibility that the drug was completely ineffective given the prior history of the response rates for the existing drug compared to the standard. Certainly, he wasn't the only person who recognized this, and I think at this point we've had guidance documents and lots of discussion and lots of papers. I think people don't really still understand the issues of non-inferiority trials very well, so I think that's a challenge. Moving on to endpoints. In the beginning, randomized clinical trials were done primarily for serious outcomes, and in some cases, intermediate outcomes were accepted to treat or to prevent such serious outcomes without people thinking too much about it. There was such a strong biological rationale. Nobody thought in trials of antihypertensives that we need to do huge studies to see whether, you know, we had fewer deaths. I mean, people looked at the blood pressure. But the consciousness was raised about 1990, one with the CAST trial and two with AIDS, where everyone was running around like chickens with their head cut off. What are we going to do about this terrible disease? And we can't possibly just wait and count bodies. We have to find something earlier. Today there's a continued push for the use of surrogate endpoints. People talk about the expense of trials. It's very, and the life-threatening illnesses without satisfactory treatments. You always have people and advocacy groups pushing and saying, you know, as soon as it's out there in phase one, we should have access to it. I think we've learned a lot about use of surrogates. I think we've learned to be cautious. I recall back when I was working in AIDS research, and there was a lot of debate about whether we could use CD4 counts or other markers as endpoints for trials, and my supervisor called me in and said, you know, this is a very vexing problem, this surrogate endpoints. You really got to resolve it. He said, I'd like you to take about 15% of your time over the next 3 months and solve this problem. You know, come up and tell us how we can be sure whether a surrogate endpoint is. I mean, that was the thinking back in those days. There must be some simple answer to this. I think we've learned that there's no simple answer. We've got to be cautious. There have been a lot of approaches proposed for validation of surrogates. I think they've been helpful in having us learn about surrogates, but I don't think there's any current consensus, and I'm not sure there ever will be on any specific way to validate in all cases a surrogate endpoint. So now I'm going to move to talking about some of the pitfalls in data analysis that we've learned about. One is exclusions from analysis. It was certainly not uncommon in early days of trials to exclude some subjects from analysis based on their failure to adhere to the study protocol, the failure of the event to meet the definitional criteria, questionable eligibility that you found post hoc after you were doing the review of all of the data after the study was done. A couple of big trials, the anterine reinfarction trial published in the late 70s and the coronary drug project, data that Dave talked about showing the problems with forgetting about the people who were non-adherent, I think raised a lot of consciousness here. And I think these papers were fundamental in establishing intention to treat analysis as a principle for analysis of clinical trials. So that's definitely a big learning factor that we had early on. Again, I'll give you an anecdote from my experience. This was about in 1980. I was working on a randomized study of adjuvant therapy for colon cancer, and this was a time when there was no accepted adjuvant therapy, and so the control arm was no treatment. It was an unblinded trial. So you either started chemotherapy, hopefully within 6 weeks of the curative surgery, or you didn't start treatment. And as we were analyzing the data, I discovered that it had been common practice. I was not around at the design of this trial. It had been common practice to cancel patients who did not start therapy within 6 weeks of surgery. It was pointed out to me, this is in the protocol. You have to start chemotherapy within 6 weeks of surgery, because why not? If you started it too late, all those nasty little micrometastases that the surgeon couldn't find, they were getting to be bigger, and you wouldn't be able to kill them with the chemotherapy. So you had to do that. So people who went beyond the 6 weeks and didn't start, they were canceled. They were not followed. We didn't have any information on them. There was no way to find out. But the people who were assigned to the observation-only arm, none of those people were canceled because they didn't start treatment in time, and I had a devil of a time trying to persuade the investigators that there was a bias here. I said, why do you think some of these people couldn't start, you know, at 6 weeks? You know, maybe they had bigger tumors, you know, maybe they were just sicker. And the person, some of you will know this person, Charles Mortel, a very eminent clinician at the Mayo Clinic, who was one of the strongest voices in cancer for good quality studies. He was the one that I was debating with on this, and one of the great prides of my life is that I was finally able to persuade him that this was a bad way to do trials. Subsets. We've always looked at subsets of interest, but we have learned, as Richard Pito has told us many times, to look at them, but not to believe the results. NIH mandates that we look at some of these. Now we've had this discussion already with the rising interest in personalized medicine. These are results in subsets are of greater interest than ever. We have learned, however, I think the Herceptin trials has been one example where the assays used to define subsets are not perfect. So people that we think we've defined who's going to benefit, maybe there's other people who do benefit, and the genetically defined risk factors seem to be much more complicated than we thought or hoped for. So, you know, I certainly am hopeful that we will get there and be able to do more of this, but I think people were overly optimistic at the beginning. A couple of my own experiences, I remember a major cancer study done by the National Surgical Adjuvant Breast Program, did a study of colorectal cancer. They showed that the experimental treatment was significantly beneficial in men and significantly harmful in women. Nobody had a clue as to what the rationale for this would be. Nobody else had studied this exact treatment. It was some kind of immunotherapy. There were other studies of similar treatments that had not shown any gender effect, but it was impossible to persuade the leaders of the group to ignore this result in designing their next study. Another example was an early trial of AZT that was done in the VA system, which showed a strong benefit in whites but potential harm in African Americans. Here, at least, we had many other studies of AZT in similar populations with no such racial difference, but it is very difficult to persuade investigators to discount their own results, even when they're faced with other results that are not consistent. When to stop a trial? Investigators always understood that it was important to watch the accumulating results and take action if it seemed necessary. In fact, they were so concerned about it that they often watched the results very rapidly as they came in and wanted to stop as soon as the p-value was less than 0.05. So we did learn that this wasn't a very good procedure. We also learned it's probably not a good idea for investigators to be monitoring their own data, and we have now independent oversight committees to do that. So while there's a general consensus that there should be some provision for stopping a trial early, nobody wants to get to the end of a trial and find that there were 200 deaths on arm A and 3 deaths on arm B. We don't really have broad consensus on the general criteria. So do we stop when the final results will almost surely show a significant difference? That's sort of the O'Brien-Fleming approach that's very commonly used in the US. Do we stop only for unexpected safety issues and never for efficacy? Do we stop early only if results are strong enough to influence standard practice? That really doesn't apply to large numbers of pharmaceutical trials that are only trying to show whether their drug is effective or not. Do we avoid early stopping because the effect estimate will be overstated? We've heard this argument from a lot of meta-analysis recently. And when should we be monitoring for futility? So these are areas of concern. So speaking of meta-analysis, the early meta-analyses of clinical trials met with substantial resistance in the medical arena. And we heard this in the cancer arena. People would say, what? You know, you're going to combine my trial, which was perfect, with that horrible trial that was done 15 years ago, and they didn't know what they were doing, and it wasn't blinded, and it was terrible. It was ridiculous. But gradually, people came to see that there was some value in doing meta-analysis of trials. But then we started learning which rocks to turn over. How could these be misled? How could we be misled? How were people searching for the trials that should go into it? What's the process of selecting studies for inclusion? What statistical methods are used? We found that there were just as many ways, if not more ways, to bias your meta-analysis than there were to bias a clinical trial. So I think we've learned that pretty much come to accept that meta-analysis can be a useful tool. I think there's some debate. Some people in the community might put meta-analysis at the very top of the evidence pyramid. Others regard this as interesting information. It's another type of observational study. Regulatory arena. We've heard a lot about the regulators today and all the horrible things that the regulators do. I think as we have learned more about what can go wrong in clinical research, we've had more and tighter requirements for trial conduct. The thalidomide disaster led to the Kefauver-Harris amendments, which included an efficacy requirement for clinical trials. I doubt that anybody would like to go back to the days when we didn't require people to show that a treatment actually worked before it went on the market. Tuskegee and other kinds of studies led to the requirements for informed consent and some IRB oversight. The Cutter incident with the polio vaccine led to tighter manufacturing controls. The Jesse Gelsinger incident increased restrictions in institutional NIH policies on conflicts of interest. I'm not sure that anybody would say we shouldn't pay attention to any of these things, or even that there shouldn't be some regulations in these areas, but a lot of us are concerned that we now have perhaps too much of a good thing. Pretty much everybody thinks that there's overkill in the regulations in many places, and there's a lot of activity now that's dedicated to pushing back where we think the regulation is creating a lot of inefficiencies and unnecessary resources. But I think we have to remember that horror stories are going to continue to arise, and when they do, they are going to get the attention of legislators who will put forward laws that the regulators will then have to have to interpret. I think of this recent example of the support trial, which was not a cluster trial and which was an individually randomized trial, but which created a lot of concern about whether the consent was appropriate. Now, do you think that that people have backed down, OHRP has backed down on that, but I think it's going to make it more difficult. The fact that that was out there in the public, it's going to make it more difficult to get traction for reducing consent in a lot of the kinds of studies we've been talking about. So what else have we learned? Randomized trials versus other kinds of research. We've learned that randomized trials of the same question don't always yield consistent results, much as we would like to believe that they would. We know that sometimes they produce the same conclusions as some observational studies, and people have written papers on how many times you got the same results from the observational study as the randomized trial. However, sometimes they produce results that differ very importantly from observational findings, and we've heard about most of these examples already. I don't know how often we would be misled if we didn't do randomized trials. I don't know what percentage of the time a good observational study would give the same results. I don't think anybody knows that, but I do worry that it's lower than we would like. So just to summarize, I think the RCT was a huge leap forward in the quest for reliable evidence. I'm not a physician, so I'm not qualified to say it was the most important medical advance of the 20th century, but certainly very high. We've learned about many ways the reliability of trial results can be undermined. We have developed methods, some clever, some very simple, to prevent many of these pitfalls, but I think we've got a lot more to learn. So thank you for your attention.